Actually, just last week there was some discussion on an email list I'm on about this very movie. It turns out that I forget how I came across it, but I sort of came across it more or less accidentally, and then it turns out that many other classes are also using this, and there was this discussion of like the places where it's more or less accurate, but it gives a really nice, the actual arrangement and the idea of how the fluid is arranged is difficult. Is much better to explain with an animation like that. I guess he didn't really say, but the point about this little lever structure is just impedance matching, right? It's that you've got some fluid, basically a transmission line here, a duct, and so the fluid has a certain, acoustic impedance ratio of its the two of the compliance in something, I don't know. The impedance, it's directly analogous to electromagnetic impedance, but it's acoustic, and it's different from that of the air. And so to make sure that the energy is successfully transmitted from here into the fluid, you have to have an impedance matcher, which is basically like a transformer. It's changing the ratio of the two properties. And so this thing is set up just so that you get efficient energy transmission from the air into the fluid. And if it wasn't the right ratio, then all the energy would just get reflected back because of the impedance change. So the way I think about the cochlea is that you can take this little spiral thing, you can unwrap it, you get something which is like this tube, which is about a little bit more than an inch long, 35 millimeters long. So it's tiny. The actual cochlear structure is tiny. And again, there's some ongoing discussion, but it's generally understood that you have something that looks like a traveling wave moving down. When you have the the motion coming in, you set up this traveling wave going down this structure, but the structure's properties change along its length. And so there'll be some point where along this kind of ribbon of, this stretched ribbon, there'll be some point where it's got where it reaches resonance at a particular frequency, and so you get different amounts of motion at different points on the membrane depending on the frequency. So there's a little animation here. Is there? Do I have it? Did I close it already? Yeah, so this is just very simple but a finite element model of how the basilar membrane works. And so basically you have the energy coming in here. This is where the sort of traveling wave starts. And so it's sort of traveling down here, but the amplitude depends on the resonant frequency of that particular part of the membrane. So the wave travels down here and it's kind of low amplitude, but then here where it's matching the resonant frequency of the structure, you get these large amplitudes, and then it's the amplitude of the displacement of the basilar membrane that causes the little hair cells to move against the tectorial membrane, which basically the mechanical stress of those hair cells being moved releases neurotransmitter and makes the hair cells fire. And so this is a high. It gets looser and heavier the further you go down. So up here it's kind of stiff and light, so it has very high resonant frequency. And down here it's sort of looser and heavier, so it has a lower resonant frequency. So you get this division of the frequency content of your traveling wave into the spatial length along the basilar membrane. This is basically the Fourier analysis, the frequency analysis that's being done by the ear, is through this sort of continuous array of mechanical resonators, but then sampled with a bunch of individual little hair cells to figure out the motion at different points along the structure. There, in the video, we saw that there are the hair cells, and then they send and they cause nerve firings, which then send nerve impulses up individual nerve fibers to the brain. There are also nerve fibers coming down from the brain to not the inner hair cells, but the outer hair cells. They're kind of like these feedback things. They look very similar, but they're, rather than firing, they're being driven. And there's an ongoing debate about what they're for, but they seem to be some kind of automatic gain control. They can modulate the sensitivity of the basilar membrane at every particular point. You know, there's a whole array of them, which would be useful if you're trying to have some kind of dynamic control of your sound sensor. Yeah?
Can you say anything about like hearing damage or like ringing?
Yeah, absolutely. So, like I said, the ear is incredibly sensitive, and it's basically these little hair cells which are the most critical part. There are these, even with all the impedance matching, it's really tiny amounts of energy that are being transmitted into the basilar membrane.
They're like analog to digital converters.
Exactly. And they have all the same problems, that they're very noise sensitive. So if you listen to loud sounds, you're basically delivering mechanical energy into these things, and eventually you can damage them, you can wear them out, you can rupture them, or you can be deafened. They're anywhere along the chain. Obviously, if you break it, it's you lose the ability to hear. But it's the inner hair cells which are usually responsible. And that's so age-related hearing loss, where you just become less sensitive to sound as you get older. That's just degeneration of the inner hair cells. And if you lose your hearing in certain regions, it's because you've lost those hair cells.
How does the ringing happen?
Yeah, don't let me forget to tell you that. Okay, so one thing about hearing loss is you typically lose if you've listened to a lot of loud sounds and damaged your ears, or if with age, you lose the high frequency first, and that kind of makes sense here because all the energy has to travel through this first bit of the cochlea, but the bit down here only gets energy that hasn't been otherwise absorbed or transmitted earlier on. So these things get a lot of wear, whereas the lower frequency end gets less wear. So typically that's why we lose the high frequency first. Ringing, tinnitus, is again not completely understood, but it seems like it's a kind of a phantom sensor situation where there are these nerves coming off here, and if the hair cell dies, then there's no information coming down the nerve. Now the brain has all kinds of adaptive schemes, and so it's trying to respond to different levels of nerve firing. And so if there's no information, then it seems like there's this again a gain control system, a different gain control system that basically pushes up the gain on that channel and says, come on, is there any information here? It's looking for information, and if there's nothing, then it starts basically hallucinating information. And so then because I don't know if you've ever experienced tinnitus, you can get it, temporarily. And it sounds like this ringing sound. Sometimes you also get this, just like, I don't even know what it is, but that you get this weird phenomenon where you just hear this sine tone. It sounds like, sort of builds up, and it'll last for a couple of minutes. That's one single inner hair cell nerve that's being somehow, it's as if that little hair cell is ringing, is firing, but it's not, because it's just some problem with a nerve in between. And so that's what it's about. But there's the origin of tinnitus is higher up in the brain, but it's because there's the lack of information coming from those nerve cells. Oh, there's also these things, these otoacoustic emissions, that because you got this. So how do you make a really, really sensitive sensor? What you can do is have a system that's on the very edge of stability, and then so that you can put in any extra energy, then it'll start oscillating, and then you can start resonating, and you can pick it up. So it seems like that there's some of that going on with the outer hair cells, and sometimes if you push it too far, then it starts ringing, starts oscillating even without input. And so there are these otoacoustic emissions where if you put an incredibly sensitive microphone in the ear, you can actually hear certain frequencies coming out, which are the actual self-oscillation of these systems. And typically, it'll be in response to an impulse that you put in a click, and then a couple of these systems will ring on for a little bit afterwards. And this is how they actually they check the hearing of newborns. You can't get a newborn to do a psychoacoustic test to say, raise your hand when you hear the sound. But you can still check this, just the first level mechanical thing, that sound is getting to the cochlea by looking for these little otoacoustic emissions. And that's really, really cool piece of medicine. Okay, so we have the hair cells. You see my picture is kind of blown up ridiculously compared to what it's really like. It's actually just this little bit down in the corner, but it's something like this. And we have the hair cells running along all the way down here, so they're all at different frequencies. There are about 3,000 of these things. So if you like, we've quantized the frequency axis into 3,000 points, so that they're all highly overlapped, right? They're not just individual things. Each of them has maybe 6 or 7 nerve fibers that they're connecting. So the actual information going from the ear to the brain is the firings on this set of 20,000 nerves. That's in humans. In other animals, the numbers change a little bit and the frequency ranges change, but it's that in mammals, that's the typical scale. And so we have the local, you can't see the plot there, but there's like a little sinusoid here which goes up, and these dotted lines show the maximum of the sinusoid. So you've got this resonant structure. It's going to move with a sinusoid around its center frequency. And then what happens is you will get the actual nerve firing. If you put an electrode on the actual nerve coming out of a particular inner hair cell, you'll see these little firing impulses. Every time it fires, it basically sends the same little shaped pulse down. But it's the time which it fires that's carrying the information, and it fires close to the center of the displacement in one direction. So again, you can't really see here, but if you plotted a histogram of the displacement of the basilar membrane versus and made a histogram of where it fired within there, it's not. It depends on the frequency, right? But it's not super accurate. So this would be like maybe 1 kilohertz resonant thing. So this is like 1 millisecond, and so it'll fire within with a few tens or hundreds of microseconds uncertainty, but it'll always fire during the positive displacement. But it won't. If it's going to fire, it'll fire in the positive displacement. It doesn't fire every cycle because these things have a limit to how rapidly they can fire, and the firing process is somewhat stochastic. But that's sort of, on average, you get this kind of behavior. But sometimes they just fire at other times as well. Some just fires because it's some noisy biological system. So you can do, we're pretty good at building electrodes which you can probe individual nerves. So you've got this bundle of 20,000 nerves, and you can stick an electrode in and record the energy coming out of one of them. Of course, it's not easy to choose a particular one. So the way that these experiments are done is you'll get your anesthetized cat or whatever, you'll expose its auditory nerve, you'll put in your little sensor, and then you'll play a range of tones, and you'll see which frequencies make this particular nerve fiber that you've got, which one makes it respond. And typically you'll get curves like this. So this is the frequency selectivity, and this is 2 different nerves that have been identified. And so we're saying, and what this is actually, what they do is they'll adjust the frequency, they'll get the nerve firing, and then they'll adjust the level of the sound until it fires at some standard rate. And so here you'll see, to make 1 kilohertz, to make this nerve fire at the standard rate, we had to put in 75 dB. But then when we got up to 7 kilohertz here, we only needed 20 dB, because basically this is the maximum frequency, the best frequency, where it's very sensitive. And if you go higher than that, right, that's saying, well, this is the energy that gets absorbed earlier in the basilar membrane. So it becomes impossible to even make this nerve fire at all. Lower frequencies, because they're going to travel sort of through this resonant point, you can make it fire with relatively, with some energy, with higher energy, but still you can still make it fire. So you have this ability that lower frequencies will work, higher frequencies won't work, but there'll be some frequency where it's much, much more sensitive. And so here's a different nerve which has a different center frequency and has a slightly different shape, but it's the same basic idea. If you put in a tone burst, then you look at where it fires, you repeat this tone burst hundreds of times and record all the firings, you get a histogram like this. the nerve is going to fire during the tone burst, and maybe if we zoomed in, we could see that it's going to fire during particular points within the cycle of a tone burst. When the tone stops, it still maybe has some sort of background rate, some spontaneous rate, but it's much lower.
This is one nerve?
This is one nerve, yeah, but the histogram pooled over multiple repetitions. But there's this onset sensitivity that when the thing starts, it's like, oh, it's really likely to fire. But then if you continue, it decays away, and then it reaches the steady state, which is, again, larger than 0, but not as much as the very beginning. And so there's a lot of auditory models try and capture this, saying, oh, maybe there's something important here that when the sound suddenly changes, then the ear puts makes a big effort to precisely represent it, but then sustained sounds get less effort to represent them. The idea is that the firing rate, because the spikes are always the same size, so how do you encode amplitude? Well, you can have it fire more often if as the sound gets louder. So here's a graph showing schematically the variation of firing rate, spikes per second, versus energy level. And for a single nerve, it's going to look like this, that there's some level below which you only get the spontaneous rate, maybe 5 or 10 spikes per second. And then there's some level where it starts increasing, then it sort of increases proportionally for a while. But then once you get to about a few hundred spikes per second, once every few milliseconds, it can't fire any faster than that. It maxes out. So you get this ceiling effect. And this range is only maybe 20 or 25 dB. Right? So that's the difference between the quietest sound that the nerve can respond to and the loudest, the most intense signal it can transmit. And so the way you can get a wider range of dynamic range sensitivity, because actually typically under good conditions, a listener can discriminate 100 dB range of sounds. One way you can do this is by having a population of nerves, some of which are less sensitive, so they don't even start responding to get to louder sounds, some of which are more sensitive, so they show you the variations for very quiet sounds, but then they saturate. Other ways you can do it is by modulating the mechanical properties of the resonator through feedback. And so it's a combination of those things that give us this very wide dynamic range as best we understand. So here's, I don't know how clearly you can see this, but here's one experiment which was done in, I think, the late 80s, where these guys at 1990, Hugh Secker-Walker and Campbell Searle, made recordings of I think a couple of hundred nerves. They found a single animal, and they went through, probing lots and lots of these different nerves in the auditory nerve just to try and get, for a single stimulus, a picture of what the nerve firings looked like over the whole range of frequencies. And so it doesn't show that well here, but basically you see these are the high frequencies, low frequencies, and the sound is a synthetic vowel, so it's got a periodicity which you can sort of see down here, and it's got different energies and different resonances, the formants, different frequencies. That's what's being indicated here, and you can sort of see that it's the different nerve fibers are picking up on different parts of that spectrum, and the overall result is pretty similar to a constant Q filter bank. I mean, that's a very, very crude approximation. But if you had to say if you had to equate this to the output of a linear filter system, then it's something like that, where you have a range of center frequencies. This is like the waveform coming in, range of center frequencies, and then different energy coming out from these different filters. And so it's which is kind of what we expect, right? The basilar membrane is a mechanical resonator. It's a bit like a bandpass filter. And then these things are just recording, halfway rectified version of what's coming out of that bandpass filter. This is a very relatively short time scale, just a few tens of milliseconds, so you can see the individual cycles of the waveform, and you can see these points of maximal excitation. So that leads to these signal processing so-called auditory models, where we try and build front-end analysis of sound, which is going to more or less duplicate what goes on in the ear, with the idea that if what we care about the sound is how it appears to listeners, what results it stimulates in the listener's brain, then maybe we want to have our computers that can have some analog of this, so we can see, we can try and control those responses directly. So that a typical auditory model will have some kind of fixed filtering to account for the outer and middle ear. Just the pinna obviously has some frequency response. And then a cochlear filter bank, which to first order is just going to be a set of fixed bandpass filters, but bandpass filters that get broader as you go up in frequency, because that's the kind of tuning we see in the ear. It's pretty much on a log frequency scale. And then some kind of inner hair cell model, which will maybe be a saturating half-wave rectification, right? So it sort of doesn't respond to negative signals, and then it responds to positive signals, but it saturates. And so here's, on a much longer time scale, the output of a model like that. So this is now, 0.6 seconds, so you can't see the individual pitch cycles. But you can see it actually, this is basically following the fundamental, and then a few other harmonics, but then above a certain frequency, the harmonics fuse together, and you just see sort of some kind of format structure. These models of these kinds have actually been pretty successful in things like speech recognition. It turns out that by having something like this in your speech recognizer, you end up with a more useful representation of sound for picking up speech, for recognizing speech. So that's a quick look at the physiology of the ear and the actual structures involved. But then you sort of get to the brain. You can say, okay, the auditory nerve is delivering this information to the brain, which has information about the different frequency components. It's already been tonotopically decomposed in different frequency ranges. It's got information about the intensity through either the use of nerves that respond to different levels and the firing rates on those nerves. But that's a long way away from, perceiving individual sources. We don't have much. It's very difficult to probe the millions and millions of cells in the brain and figure out what's going on. And so at this point, experimentation tends to switch to psychological, which is you take the entire organism and you try and see how it responds. And in the case of human psychophysics, one of the things you can do is you can just ask people to do tasks, play people sounds, ask them questions, and see what they come up with. And then you end up with this, you can start probing the relationship between physical parameters like the frequency of a sound in cycles per second, the amplitude of a sound in dB relative to some reference energy level, and the psychological values, which is what pitch someone says they hear and how loud they report it as being. And this is, we tend to think of these things as the same, right? That there's a physical parameter which we directly perceive, but of course they're not the same. There is a gap there, and so there's quite a lot of care that has to be taken to correctly understand exactly, to describe and understand how that relationship works. Hearing is, of course, not the only place this comes up. It comes up in vision. There's all kinds of weird psychovisual effects, like, color balance. You adapt to the local lighting to figure out the color of things. And then even things like, perception of weight and other kinds of perception of body position, things like that. Again, this is something that, as an engineer, I didn't know when I first started in this field. So here's a classic example, which is the perception of loudness. One of the ways you can measure this is by looking at the just noticeable difference, which is the smallest change you can make that a listener can reliably identify as being a change. And typically, you would. Typically, the way you do this is with paired tests. So you play people two stimuli, and you say one of these two stimuli has a change in it, or has, one of them the frequency increases, or maybe the frequency gets louder, or the sound gets louder or gets quieter. Which one is it? And people say, well, they sounded the same to me because they're right on the very limit of perception. But if you force me to choose, then okay, I'll say this one. And it turns out that even though they're reporting that they can't tell, they are actually able to tell. And there's this interesting kind of conscious filter, like the consciousness says, well, I can't really tell, so I'm not going to give you an answer, but yeah, if you really want to know, it's this one. the kinds of results you get here, in fact, over very many physical parameters, are that the just noticeable difference change is proportional to the absolute level, which means that as things get louder, you have to change them by a larger amount before you can notice them, but by the same proportion, right? So it's always like you're sensitive to like a 1% change or something like this. And what that means mathematically is that there's a logarithmic proportionality, that if the perceived level is represented by L and the physical level is represented by I, then these things should be, if you could quantify this, they should be related they should be linearly related on a logarithmic plot. But the constant of proportionality depends, right? And so it turns out that for loudness, when you try and do this, you get a constant proportionality of 0.3, which means that, or when you express it in direct times, you get this power law relationship. That the perceived loudness, if you make people put a number on it, is proportional to the intensity, the energy level, the amplitude rather, to the power of 0.3. I was going to run this experiment actually for real. I don't think we've got time to do it now, so we'll skip it. But let me just play you the stimulus. So basically, the experiment is you get a sheet like this with a set of numbers, and you write down, you say, okay, reference sound is 100. How loud was that? And it's like, let me play the example, so you can see what that sounds like. So,
loudness scaling. In this experiment, we will rate the loudness of 20 noise samples, which are preceded by a fixed reference. First, you hear the reference sound, followed by the strongest and weakest noise samples.
Okay, so that's like, there's a reference which is 100. Then you hear something louder and you have to give it a number. Then you hear something quieter and you have to give it a number. And so
now, the 20 samples. For each sample, write down a number reflecting its loudness relative to the reference.
So that one's much louder. What number would you write down? 200, yeah. 200 is popular. And then, so here's another one. That sounds kind of the same, right? Sounds a little bit quieter. Okay. I mean, so it's the amazing thing is that it's like, what do you mean I have to write down a number? But people will do it, and people will do it relatively consistently. So when I have done this in class before, and these are actual results that we got from a classroom experiment a couple of years ago. And basically, there are some each of the sounds is at one of these levels, and then you take the log of the response. So 100 will be 2, right? Log base 10 is here, so that's the reference. And you get this plot, and I'm sorry, the contrast's not very good, but this cyan line you can see here is the 0.3 slope, so a change in 10 dB gives you a 0.3 change in the log of the mapping, and the lines fall quite well onto this. These are the actual data points, and they fall pretty well. So you can do this experiment for live on real results, and you get kind of what you expect here. But that's kind of the nature of how we get information about perception, right? We do experiments like that where we make people do these kind of weird things. And if you set up the experiment right, if you control for confounds, and if you do it with enough people, then you get a result which is repeatable and and reliable, that you do actually get measurable. You can measure the perceptual system. Yeah. One of the one thing you can do is to try and measure how people map energy level to perceived loudness as a function of frequency by playing them two different frequencies and saying, okay, adjust the energy of these, adjust the level, here's a volume knob, adjust them so they're the same intensity. And then what you find is, and this line here is the level that people will adjust it to as a function of frequency to match a 1 kilohertz tone at a particular energy. And these are like, the 1 kilohertz tones are like 5 dB apart or 10 dB apart here. And you see that it's not quite flat, that people are actually a little more sensitive to 4 kilohertz than to 1 kilohertz, which is why smoke alarms use this frequency, 4 kilohertz. But then as you go up, you suddenly become much less sensitive above around 15 kilohertz. And you're gently less sensitive at lower frequencies down to about 100 or 50 hertz. What's interesting here is this compression of lines here is saying that, well, at 90 dB, you got fairly flat sensitivity, right? So 90 dB at 100 hertz sounds loud, and it sounds about as loud as 90 dB at 1 kilohertz. But at at 20 dB, you can hear a 1 kilohertz tone pretty well, but you can't hear a 100 hertz tone at all, right? Its loudness is below the quietest. This is like the quietest sound you can hear. And in fact, you have to raise it to like 40 or 50 dB before it even sounds that quiet. And so there's this kind of compression of the loudness scale that suddenly, what takes 60 or 70 dB of energy change to give you this range of perceived loudnesses at 1 kilohertz is now only, 40 dB. It's only half a change. So you have this perceived expansion of loudness. Yeah.
So how would like a 12-year-old kid's curve be? Like how?
So the age is all about the high end, right? So basically, this is probably an average adult. A young kid has a much extended range up here, and then an older person, this thing pulls back until it starts getting into this range, which is like the key. I guess this range is the key speech perception range. So once you start losing some of this, then you really can't understand so well.
And it doesn't really matter in low frequencies?
The low frequency stuff, it doesn't change, I don't think, that much. But what happens when you do have hearing damage is this compression sort of gets, these lines get pushed up, right? And so you have the same effect where actually sounds rapidly become very loud. And so one of the problems with hearing loss is that, you can amplify the sounds with a hearing aid, but then it's very easy to make them uncomfortably loud because suddenly you're pushing them up into this range, which is where your brain starts saying, you're damaging your ears, you've got to back off. This is kind of, this is a detail of hearing perception, not a big deal in everyday life. But it just makes the point that the things that we assume we're doing right, which is like, well, obviously we have this continuous proportionality of our perception to the physical property. Turns out that there are these limitations to what the sensor can do, right? This is basically, it's just difficult to get that much energy in these low frequencies, so we can't perceive as much energy at these lower frequencies. But the brain sort of tries to make it up by expanding things out, but there are definite limitations to what it can do. One of the kind of, again, these limitations that you're not aware of comes from masking, which is basically, because of the dynamic range limitations, if you have a loud sound next to a quiet sound, then it's much harder to hear the quiet sound than if the quiet sound is there on its own. Because basically they're going into the same sensor, and the sensor has a certain amount of noise. You can have some automatic gain control moves it up and down, but if the AGC is being tuned for this high-level thing, then you can't pick up the low-level thing. And so this is like this lowest curve from the Fletcher-Munson curve we saw in the previous slide. This is the quietest sound you can perceive, and we can put tones in here, and if it goes above this curve, you should be able to tell that it's there. So this would be, you can just see this point here. Normally this tone would be perfectly perceivable, but if you put a steady tone in here, a masking tone, and you sort of do the same thing of putting in energy at different frequencies and seeing how loud you have to make it before someone can tell where it is, you get this modification to the masking curve like this. So it's like you've become locally less sensitive in the region of this frequency, which is basically because, you know, those nerves are already doing stuff. And so this tone then becomes inaudible. You can't tell whether it's there or not. And this is actually used in psychoacoustic data compression like MPEG audio. They break up the frequency into separate bands like this. They separately quantize each band, and the actual amount of quantization you have to use within a band, which is basically the gap between the signal and the quantization noise, it becomes much lower because now you only have to quantize to make sure your noise is below this masking skirt here, rather than, you know, if you had, because quantization noise comes in flat. If we had to make it so that it was flat everywhere, then we'd have to make it much lower to make it work. So that's simultaneous masking, and there's also sort of a forward masking in time. So here, this is like time versus frequency versus masking level. If you have a masking tone like this, it'll have these kind of these shoulders coming from simultaneous masking, but even after it's stopped, it takes a while before your sensitivity recovers in that frequency. So you have this forward masking skirt, which is typically 100 milliseconds or longer. It's like, you know, it's meaningfully long in perceived time. And you can also use that, again, for improving compression, for reducing the amount of information you have to send in a sound without changing how it's perceived. So roughly speaking, you know, you can do all these kinds of experiments where you play people two references, play them a third sound, ask them which one it was. Again, you know, two alternative forced choice. And you can see what are the things, what are the levels below which people can't tell the difference between sounds anymore. In timing, we're pretty sensitive to really small time. 2 milliseconds is, you know, the blink of an eye, much less than the blink of an eye. But if you, under certain structures, you can, like if you take like a synthesized music performance and you start messing with the times of the events, once you get to a few milliseconds, people begin to say that, you know, that's not quite right. It doesn't sound quite right. If you actually play people two sounds and ask them which one is first, then they can't tell at 2 milliseconds, but at 20 milliseconds or so, they can sort of tell which one is first, right? That's when you begin to be able to actually hear time as time. In terms of pitch tuning, a semitone, right, the gap between the two closest notes on a piano is about 6%, right? Because that's the 12th root of 2 is 1.06 roughly. You know, when you're tuning a guitar or something, you can detect tunings much smaller than that. Best case, your tuning sensitivity is about 1%. And probably a trained musician can do better than that. But typically, if it's within a couple of percent, it doesn't sound out of tune. But that's about the limit. In terms of loudness, there are tons of experiments that have been done where they'll play people a set of sine tones, then adjust the energy of individual ones by small amounts and see how they can tell. And your sensitivity is around a couple of dB there. The experiments we did with the experiments we were talking about doing with loudness means that actually a change of around 9 or 10 dB is perceived as a doubling of loudness. So this is like, you know, maybe a fifth of that is the limit of what you can detect. Yeah.
So how is tuning put into units? Like you said, 6% is
yeah.
half tone. So
This is in hertz, right? So that if I play you 1,000 hertz tone, and then I play you like 1,005 hertz tone, you wouldn't really be able to say whether it was higher or lower in frequency. But if I play you 1,010 hertz, then that's when you can say, okay, that's higher in frequency. But but for 100 hertz, your sensitivity is not so good at 100 hertz. But if it was the same, then you'd be able to tell 101 hertz, a 1 hertz difference at 100 hertz. So these are things we're pretty good at picking out. Some things that we're not so good at picking out, we're not very sensitive to phase. It's not even clear what you mean by phase. But if I play you two sinusoids that are multiples of each other, right? So I could play them in sine phase, so they both go through 0 at the same time. Then maybe this one does 3 cycles, this one does 1 cycle. I could shift the phase of this one, so now instead of going through 0, it goes through its maximum. you would not be able to tell those apart because they go into separate, you're quite good at recognizing that if you shift the phase, it's the same. It sounds like the same pair of sine tones. There are different, if they were going into different ears, you'd be able to tell them apart because then the phase shift would correspond to a time shift. If they were the same frequency, then you hear a phase shift and you're very, very sensitive there. You're sensitive to like a few tens of microseconds in phase shift there. But in general, if you take a sound and you mess with its phase, it's doesn't change much. Of course, we're very good at recognizing these sine tones and hearing the different frequencies, but another kind of sound is like a noise sound, like white noise. You can think of that as a collection of a very large number of sine tones. But if I play you two different noise signals, but they're the same average spectrum and the same energy, you can't tell them apart, right? It just sounds like noise repeated. Interestingly, if I take a noise signal and I repeat it over and over again, I loop it, like maybe 500 milliseconds of noise and loop it over, then after a few seconds, you begin to hear something. You hear these sort of whooshing sounds, you hear the periodicity, you notice that it's repeating. And then you would be able to tell the different noise tones apart. But that's kind of not fully understood. It's like one of these automatic gain control pushing, really magnifying out to try and pick out the features. Although people are fascinated with that, and they've done experiments to see whether different people hear the same things, and they don't, and it's all very complicated. But and also it turns out that birds are much better at it than we are. They don't hear noises as equivalent in the same way that we do. But that's sort of roughly where we are with what people can and can't tell. Okay, so we have a few minutes left. Let me talk a little bit about pitch perception, just because, again, it's an interesting case study of something that's relatively clearly defined, something we do very directly, and we don't really think that it's such a big deal. But when you try and figure it out, it turns out that it's more complicated than it seems. So if I have a signal like this, right, so this is just a bunch of alternating impulses in time. It's like the sound that came out of the plucked string. This is actually the output of a plucked string algorithm, a Carper-Strung algorithm. You hear a pitch, right? You say, oh, that's a C, or that, you can match the particular sound. You could hum the sound back. You could hum the note back. Even though the sound you are humming is not the same sound, right? It's a different waveform, but it would have the same pitch. You'd perceive it as having the same pitch. You can match different waveforms according to their pitch. So there has to be some mechanism in the brain where that pitch is calculated and represented. And there are a couple of things we could do. Firstly, this is the output of a constant Q filter bank, and it's again over a short time scale, so you can actually see the output of the individual cycles coming through here. Now, this is a complex tone, so it has this Fourier spectrum which has got F0, 2F0, 3F0, 4F0. One thing you could do, and it's not the worst thing you could do, is just look for the lowest peak and say, well, that's going to be F0. The lowest harmonic is always at the fundamental, sort of always. There are going to be these higher harmonics. We just ignore those, look for the lowest one, and report that as the pitch. It turns out you can filter this out, or you can mask it with noise, and people still hear the pitch fine. So it's not the fundamental that's critical, although it's certainly, if you play the fundamental alone, you hear the right pitch. You just play a sine tone, you match the pitch. So the simplest thing isn't explanatory, and also that experiment shows that the model itself isn't a single thing, because I can take this sound, just the higher harmonics, or this sound, just the low harmonic, they're non-overlapping sounds, and you can still get the same pitch. So the pitch has to be coming from a more complex calculation. So the two competing theories are place models and time models. In a place model, you have maybe this profile of excitation on the basilar membranes. This is the frequency channel, this is how intense it is, and here are the individual harmonics. We know that we don't need any particular individual harmonic, but if we have a subset of these, we do hear a pitch. And so what you can imagine is some kind of template, sometimes called the harmonic sieve, where you say, well, if I have this particular pattern of harmonics, if I have a lot of energy consistent with this pattern, then I think that pitch is probably present. Although I'm not saying there's no single part of that template that has to be present. So just, I'm going to calculate the inner product against this whole template, and then if I have a lot of match, then that'll be evidence for that pitch. If you have this is not physiologically relevant, but from a computational point of view, if you plot this on a log scale, then this is always f0, 2f0, 3f0, 4f0. On a log scale, there will be fixed distances, right? Because the ratio turns into a fixed distance. So you can have a sieve like this, and you can just do a cross correlation. You get something like this. So now you have pitch strength as a function of frequency. And here, for this nice harmonic pattern, you see this big peak here. And in fact, even though there's a little bit of a peak around here when this spike lines up with here, it sort of gets washed out. You're not confused by this harmonic to think there's a sound octave above, which is quite consistent with our perception that even when you hear a harmonically rich tone, it's really hard to hear the pitches associated with the individual harmonics. But there are a bunch of signals you can make which don't really give excitation patterns like this, but do still give a strong pitch. If we basically put in a lot of noise components, we could make this approximately flat. But if we introduce some periodicity to those noise components, we can still have a perception of pitch. So that supports more of this time model, which is saying it doesn't matter where in the frequency axis you get this energy. What matters is the fact that a particular period will always have a periodic structure. Either it's because it's the actual period of the frequency itself, or up here it's sort of the envelope of the signal. You can extract that periodic structure by doing an autocorrelation of the energy of the envelope of the energy in particular frequency bands across all the different frequencies, and you can end up with this so-called correlogram, which is frequency channel by lag, and then it varies as a function of time. And you can have the so-called summary autocorrelation, where you sum up across all these channels, and you get this evidence for periodicities at different periods. This is a kind of when I first learned about this was very appealing because it's a unified system. It works for sinusoids, but it also works for higher frequency energy that's modulated at the same periodicity. Does some weird stuff in between, depending on how you do your rectification and things like that. But even here, which is the second harmonic, so we have modulation, which is at twice the frequency. If there's a fundamental there, then it's still going to reinforce the fundamental. Problems with this is that it's not a terribly good predictor of pitch phenomena, not in detail. Here we'd predict that if we only take the 10th harmonic and above, but still have this, if we take a complex sound and then filter it out to only keep the higher frequency energy, we'd still have a strong pitch percept. It turns out that if you don't have any of these resolved harmonics, you still get some pitch percept, but it's pretty weak. That there is, your ear wants to see these individual resolved harmonics in lower frequency. So it seems like that if you wanted to build a really accurate model of how people perceive pitch, you need to have both of these principles in use. And that kind of makes sense, again, from an evolutionary point of view. The brain doesn't care whether it's using like a single, very nicely principled way of detecting pitch, or whether it's sort of using a messy combination of a bunch of different things. It just cares about being as robust as possible. And so it seems like with this, as with a lot of perceptual phenomena, the way the perceptual system works is it has a bunch of different cues it can use, and then does some kind of evidence integration. And of course, we have a nice theoretical framework for that, the sort of the rigorous rules of probability. That if we're trying to figure out a particular parameter or attribute like theta, given some evidence, x, some observations, then we're trying to find the theta that maximizes the posterior probability. Then if we have two divisions of this evidence which are independent, then we can basically work out the probability under each of these subsets of the evidence and multiply them together. There's some normalization you have to do, but this would be, the actual what Bayesian probability theory would tell us to do. And so you can imagine this as like we have an estimator of the pitch based on the spectral profile, the place model, an estimation of the pitch based on the autocorrelation, and we multiply them together to get an overall map of different likelihoods of different pitches. And then if the place information is weak, the timing information will dominate. If the timing information is ambiguous, then the place information can dominate. And if they're both strong, you can get a stronger percept. And so, again, it's not obvious how you implement this in a neural scheme, but there are a surprising number of perceptual problems where the solution seems to be quite the performance of listeners is quite close to what you would consider a Bayesian optimal analysis. That it really is doing the best job of analyzing the different information available to give you the best possible result. Although it's always interesting to find the places where the system breaks down and doesn't give you the best information, which is what gives you illusions and things like that.